Hello my dear students and welcome back to Excellence Badge, and I am your Diksha ma'am. So today we are going to start with the topic fertilization. So after we have discussed about the male and female reproductive system and gametogenesis, so the next step is the fertilization, because that's the motive of gametes, right? In organisms that are sexually reproducing, why do they produce gamete? Because one day those gametes will meet, they will fuse, and then they will do development and make a baby or the next generation. So in the starting of the chapter we have done little processes, right? How the sexual reproduction in human take place. First there is like gametogenesis, then there is insemination, and then there is fertilization and so on. So we are going through that process only. So gametes have been formed. Now let's see how the fertilization takes place. So for that, let's begin fertilization. First of all, what is a fertilization? What is fertilization? It is a fusion of haploid means N, haploid male and female gamete, haploid male and female gamete to form a zygote. To form a zygote which is diploid, right? So this is the definition of the fertilization, that two gametes, like what are these two gametes? Like this is ovum or egg and this is sperm. So sperm is fertilizing the ovum, and later on what it will become? It will become 2N zygote. Fine, and this zygote will then become embryo, and then a baby will come out. After embryo, what is formed? Fetus. Embryo is that stage, early stages in the womb of the mother, and fetus is a baby which has all the organs that has been formed, right? And when it will take birth, you call it as infant. After birth, the baby is known as infant. That's the difference. Okay. Now, is this fertilization so simple? So what do you think so? If the fertilization is so simple, why not everyone have a lot of babies in their life? Though female have 60 to 80,000 follicles, why can't we have 60 to 80,000 of the babies? Though some have a lot of babies, like recently I heard a news where a female gave birth to like 11 or 12 kids in, I think in the US or so, I don't remember, right? So why can't we just produce 60 to 80,000 of babies? Because there is a limit to things. So this fertilization is not the always successful process. Sometimes it is unsuccessful also. So let's go into much detail. So for fertilization, there should be first step that should definitely take place, and that is insemination. That is insemination. What is insemination? Whenever male and female undergo sexual intercourse, male deposit semen inside female's vagina, right? So for example, this is uterus, this is cervix, okay, and this is vagina. So male will deposit semen in the vagina here during sexual intercourse. Imagine this is semen, this is vagina. And you also know, per ejaculation, male releases around 200 to 300 million sperm. So we are assuming the male is healthy, so this semen will be carrying 200 to 300 million sperms. Okay, so this is the first step for fertilization, that is insemination. What happens in insemination? The male deposit semen in the vagina of female. That's the first step for fertilization. If this does not take place, fertilization will not take place, right? Next, what will happen after that? After insemination, this contains a number of sperms, right? Now, these sperms, they have to reach at a place known as fallopian tube. This is fallopian tube, this is ovary. Okay, so sperm have to reach at this place known as ampulla, because ampulla is the site of fertilization, right? These sperm in the vagina, they have to reach at this place. Okay, so now the sperm need to run, they need to run. So if sperm need to reach at this place, there are certain factors which will allow the ascent of sperm, or which will allow the sperm to move up. First thing will be the vacuum created by the uterus. This is uterus. So uterus and vagina's positioning is such that the uterus is creating vacuum pump, right? As a result, whatever will be in the vagina, it will suck it. It will suck it. So for ascent of sperm, there are certain factors which are responsible. Ascent of sperm means, ascent means to move up. So for ascent of sperm, the first thing is the vacuum created by uterus. Vacuum created by uterus. Second thing, during sexual intercourse, the female's vagina and uterus, they undergo muscle contractions. So contraction of the uterus and vagina, they will also allow the sperm to move up. So second thing that is responsible for ascent of sperm is contraction of uterus and vagina. Contraction of uterus and vagina. And third, very most important is, you all know the sperms have this tail which is like a flagella, right? So this will help the sperm to swim. So yes, the movement, the swimming movement done by the sperm is known as whiplash movement. You know what is whiplash? So you have seen in movies, in Hindi you call it as kode. Right, so whiplash is some long whip, you must have heard of whip, right? So it's a long rod like structure, it's like a rope also, you can say. It's tapering at the ends. So they used to, you know, beat people with that whip. So just like how the whip moves, they also show that kind of a movement. So that's why we call it as whiplash movement. Whiplash movement of sperm. So all these factors will allow the sperm to move up. So you can see the sperms are running. So only 100 to 200 sperms reach up to here. Only 100 to 200 sperm out of 200 to 300 million sperm, only 100 to 200 sperms reaches the fallopian tube. Now in the female genital tract, when the sperm enters in the female genital tract, the sperm undergo changes, right? When a sperm is in the male's reproductive tract, sperm is literally inactive. It is literally inactive. But when it reaches the female reproductive tract, sperm need to get activated. Because if sperm will not be activated, it will not show the whiplash movement. It will not show the whiplash movement, right? So that activation of the sperm is known as capacitation. So capacitation are the changes that occurs in the sperms when it reaches female genital tract. Female genital tract. Right, got it? So there are certain changes that will occur in the sperm. Now what are these changes? For example, this is the head of a sperm, this is the neck, middle piece and tail. Fine. Now we need sperm to move very fast. So the first change that will occur, that calcium will start to enter inside the sperm. Because if calcium will enter, it will allow the fast movement of the sperm. If calcium will not be there, sperm will not be able to run, right? So the first change that occur is entry of calcium ions. For vigorous motility. Second change, you all know in the head portion of sperm, the nucleus is present and above that acrosome is present. An acrosome contains enzyme for digesting the wall of the oocyte, right? So we want that now it's time to digest the wall of the oocyte. Why not to free the acrosome? So to free the acrosome, there are certain certain cholesterols present on its membrane. Imagine this is a plasma membrane, right? And what are these cholesterol? So when I will remove the cholesterol from this membrane, the membrane will disintegrate, the membrane will break, and if the membrane will break, the acrosome will be exposed, So that's the second change that occur. Cholesterol is removed, to break the plasma membrane and expose acrosome. Fine. And third very important change, when a sperm is in the male reproductive tract, it is, I have told you, it is quite of an inactive stage, right? So what makes it inactive? So when sperm is passing through epididymis, epididymis secretes some decapacitation factors. What factors? Decapacitation factors. So these decapacitation factors are present on the surface of the sperm. As a result, a sperm is not able to do all these changes, right? So what are these? Decapacitation factor. These decapacitation factors will make sure that capacitation should not take place. These two changes should not take place. So the very first thing that happens when it enters female reproductive tract, it will remove its decapacitation factor, because when it will remove its decapacitation factors, all these things can easily happen, and these factors are given by epididymis. Because if if the sperms it gets mature in the male reproductive tract, then it will be useless. Why? Because after capacitation, the sperm's lifespan is just 48 hours, and you never know when male is going to ejaculate the sperm. If the capacitation would have taken in the male reproductive tract, after 48 hours the sperm will be useless, right? So you don't want sperm to get useless or to get destroyed. So nature has given the opportunity, why not to make capacitation in female tract, because then after that fertilization will take place, right? So epididymis give this decapacitation factors. So removal of decapacitation factor is the first step in capacitation, right? Because after capacitation, what will be the lifespan of sperm in female tract? Lifespan of sperm in female tract, I have just told you, that is 48 hours. Right? So that's why, like if someone has done copulation or sexual intercourse today, so for around 48 hours, like 2 days, the sperm can easily survive in the female tract, right? Now this is the thing that the changes that should occur in the sperm. Moving further to the next. Now what will happen next? So we say this is the uterus, right? This is the fallopian tube. This is the fallopian tube. Now sperms, they are rushing towards the ampulla. Okay. Now we say the fertilization always occur at the ampulla. But do all copulation or sexual intercourse lead to fertilization? No, that's not possible. It's not always, it's a probability. Might be the first sexual intercourse unprotected, right? The first sexual intercourse might lead to to fertilization, but there can also be chances like even after 12 or 13 unprotected sexual intercourse, the fertilization will not take place. So having a baby or the fertilization is a chance process. Why? Because there is a condition given by nature. What is that condition? The condition is only fertilization will take place only if the sperm and oocyte the sperm and oocyte will reach ampulla simultaneously. If at the same time, at same place they will reach together, only then fertilization take place. Any of them is late, fertilization will not take place, right? So it's a chance process. For example, the sperms were reaching at the same time, ovary had done ovulation. After ovulation, the ovum also have reached at the ampulla, like this. Now what will happen? Any of the sperm will fertilize it. So we say that yes, the site of fertilization is ampulla, but fertilization will only be successful if both sperm and oocyte, they reach the ampulla simultaneously. Simultaneously means both at same time, at same place they will be reaching, right? So we say fertilization takes place in ampulla when both sperm and ovum, now do not mislead it, both sperm doesn't mean two sperms, both we are talking about sperm and oocyte, right? Both sperm and oocyte reaches ampulla simultaneously. Alright, now when you will be solving the previous year question, the question that has been asked, what is the site of fertilization, might be you will find the option not ampulla but ampullary isthmic junction. In the previous NCERT, it was written that fertilization takes place at ampullary isthmic junction, but now it has been changed. The truth is fertilization takes place at ampulla. So do not get misleaded. Do remember, if you will get the question, mark ampulla, the site of fertilization. Okay, all right. Now moving further. So is it that easier that fertilization is taking place? Okay, the sperm and ova are meeting. So is that it? This is how they meet? No. Behind even their fertilization, a lot of physical chemical process take place, right? So we say fertilization is not just meeting of sperm and ova. Rather, fertilization is physico-chemical process. Fertilization is a physico-chemical process because physical factors and chemical factors, they both play a very important role. So first we are going to talk about this chemical reaction known as acrosomal reaction that takes place before fertilization, right? Now oocyte that has been released, it looks like this. The oocyte that is released from the ovum, it is not just single cell. You all know it also contains a lot of layers around it. It also contains a lot of layers around it, if you remember, right? So there is an outer layer of glycoprotein known as zona pellucida, and then there is a layer of cells known as corona radiata. This is what is released from the ovary and this is what enters into the fallopian tube, right? So this is secondary oocyte, and you all know this secondary oocyte has started meiosis 1 but it has not completed it. It is at halt of metaphase 2, right? So here metaphase 2 not completed. Meiosis 2 not completed, it is at halt. Halt where? Metaphase 2. Alright. And this layer, the orange one, is zona pellucida, which is made up of only glycoproteins secreted by secondary oocyte. It does not contain any cells, and this is corona radiata. Corona radiata is the layer of the follicular cells, and these cells, they are bound to each other. They are bound to each other with the help of a sticky substance, and this sticky substance is known as hyaluronic acid. What is this? Hyaluronic acid. So in between the cells of corona radiata, this hyaluronic acid is present. What is hyaluronic acid? Hyaluronic acid is basically a mucopolysaccharide. It's a modified sugar. You found it in, or you can find this in aloe vera gel, in the okra, ladyfinger. In ladyfinger there is a sticky substance. So this is a sticky kind of substance. It is acting as a glue between the cells of corona radiata, right? Now imagine in that race of sperms, any one sperm, in this race of sperm, this color is nice, in the race of sperm, any one sperm is the one which has won the race, and it is the one that has reached the ovum at first. Imagine this is the sperm that reached the oocyte first. This oocyte is bigger, ovum is, the sperm is very small, right? Now this sperm has to penetrate inside the oocyte. How it will do? It will start bursting its acrosome. That's why its name is acrosomal reaction. It's a chemical reaction where acrosome will release its enzyme. Acrosome will release its enzyme. Why? To penetrate oocyte. To penetrate oocyte. Right? So now what enzyme it will secrete? There are certain enzymes. The first enzyme that this acrosome will release is hyaluronidase. Hyaluronidase, ase is always used for enzyme, and most of the enzymes name is on their substrate, right? So this hyaluronidase will digest the hyaluronic acid, which is like a glue present inside the cells of corona radiata, because the first layer that you need to penetrate is corona radiata. So first we have to digest that, and there are two things that we need to digest. First, the cells of corona radiata, and second, hyaluronic acid. So first enzyme, hyaluronidase, what it will do? It will digest hyaluronic acid. It will digest hyaluronic acid. And second enzyme that it will use is CPE, corona penetrating enzyme. Corona penetrating enzyme. And this enzyme will help in penetrating cells of corona radiata. Helps in penetrating. Penetrating means to enter. Penetrating cells of corona radiata. Alright. So it has digested the corona radiata. It has entered more inside. Okay. It has entered more towards inside. Imagine it was here. Now it has entered somewhat here on the zona pellucida. So as soon as it will reach the zona pellucida is not that easy to go. First of all, you have to exchange the passwords. First of all, you have to exchange the password. Why there is a password? Because we want the sperm, which is the human sperm, should only penetrate the human oocyte. This sperm, if it is of another species, of another animal, the zona pellucida will not allow it to enter. So that's why it is asking the password. And what is a password? Password is in the form of proteins. There are certain protein receptors present on the surface of zona pellucida. If they match with the surface protein of sperm, only then the sperm will be allowed to move inside. If they do not matches, they will not be able to move inside. Let me show you how. For example, this is zona pellucida. Okay. And these were the cells of corona radiata. Okay. So sperm here has entered. Now there are certain proteins present on the zona pellucida. So these protein, if they matches with the protein of the sperm, these protein if they matches with the protein of the sperm, only then sperm can enter inside. So there are surface protein present on zona pellucida. If they matches, only then the sperm can enter inside. And hence we say that our fertilization is species specific, that only same species gametes can fuse. If the gametes are of different species, they will not be able to fuse, right? So here very important point, fertilization is species specific. How? Now what are the name of the proteins present on the zona pellucida? ZP receptors, right? So ZP receptors on zona pellucida should matches with sperm protein, right? Now the match has been occurred, but it's a thick layer. The zona pellucida is a thick layer of glycoprotein. We need to penetrate it. Also. Okay, let me just demarcate it like that. We need to penetrate it. So for penetrating zona pellucida, there is third enzyme known as acrosin, or zonalysin. Once the recognition has taken place successfully, once ZP receptor has matched with the sperm, now the acrosomes enzyme, which one, acrosin or zonalysin, will be released. This will digest the glycoprotein of zona pellucida and sperm will enter more inside. So now what will happen after this, guys? This is the enzyme. Okay, zonalysin. This is the enzyme to penetrate zona pellucida. Fine? Okay. Alright. So let me draw the diagram one more time. So that's your oocyte. And this is your zona pellucida. Alright, and this was your corona radiata. So now sperm has entered here. So this membrane or plasma membrane of oocyte, it has a name. What you call it as? You call it as vitelline membrane. So vitelline membrane is the plasma membrane of oocyte. What is it? Plasma membrane of oocyte. Now sperm has entered on the plasma membrane. So again on this plasma membrane, the proteins will be checked, right? So here there are proteins known as fertilization and anti-fertilization. Fertilization is present on the surface of vitelline membrane, right, this one, plasma membrane of your oocyte, and anti-fertilizing protein is present on the surface of sperm. If they both will recognize each other, the sperm will get an entry inside. Just like if you have a key, only then you can open the lock, right? So imagine the vitelline membrane or the oocyte membrane, it has a protein known as fertilizing, and sperm has the key, and what's that key? Anti-fertilizing. If they both recognize each other, only then the sperm can enter more inside, and hence the sperm will enter towards the cytoplasm. So that's events are very much important, and they are acrosomal reaction, because these are the enzymes that are coming out from the acrosome. So that's why you call it as an acrosome reaction. Fine. Okay. Now what happened next? Let's see. So here this is vitelline membrane, this is oocyte, this is zona pellucida, right? Now sperm has entered inside the cytoplasm. Okay, now you can see the sperm has entered inside the cytoplasm. So as soon as sperm touches the cytoplasm, the metabolism of the egg, it enhances. Or you can say earlier the oocyte was sleeping. Now oocyte, when it got touched by the sperm, or oocyte or the cytoplasm of the oocyte gets touched by the sperm, it will wake up, right? So when the sperm enters the cytoplasm, the metabolism of the oocyte cytoplasm, it will gets on, or it gets enhanced, or it starts, and it was at metaphase 2. It was stuck at metaphase 2. So as soon as it will enter, the sperm will enter the cytoplasm, the metaphase promoting factors, they will diminish, they get destroyed. And you all know after metaphase, anaphase comes, right? And now to enter into anaphase, we need anaphase promoting factors, right? Or anaphase promoting complex, APC. So now if you want that here your oocyte was stuck at metaphase, so we want to enter into anaphase, we need proteins of anaphase, right? So when the cytoplasm gets touched by sperm or sperm enter into the cytoplasm, metaphase promoting factors diminish and anaphase promoting complex, it enhances or increase in number. As a result, the meiosis 2 will be completed and this egg, oocyte, it will complete its meiosis to form ovum and a polar body. So you can see here when it will touch the cytoplasm, I am writing here also, sperm enters cytoplasm of oocyte. Now what will happen? What will happen next? When it will enter here, the two cells will be formed. Two cells will be formed. This is polar body. This is second polar body, right? And this is now ovum. This is now ovum. Another point, how does it enter from metaphase to anaphase? Here, metaphase promoting factors MPF, they will decrease, whereas anaphase promoting complex APC, it will enhance, right? Short form is MPF APC, and this is how it will complete its meiosis 2, right? And second polar body will then later on degenerate. Now a very important point to note here is that, for example, this was the vitelline membrane, okay, and sperm was entering. So the place from where the sperm enters, in the oocyte, that becomes swollen, or we can say the place from where the sperm is entering, that becomes swollen, because now oocyte has also somewhere got a stimulus but that sperm is entering. So it is making a space for sperm to enter. Okay, so whenever a sperm reaches the vitelline membrane, the oocyte produces this swelling like structure known as cone of reception. And once from cone of reception it enter inside the cytoplasm, meiosis 2 will be completed, and now ovum has become haploid, okay, because polar body is also haploid. Now sperm will only give two things to the ovum. One is its nucleus, and second is its proximal centriole. Because ovum has its own centriole, because it's just one. It does not have the second centriole to complete the divisions. Because when you want to complete the division, you want to make a metaphasic plate and spindles. For example, there are chromosomes present, right? And you need to make spindles like that. So for that you need what? Centriole. So this is the centriole of ovum or egg, and this is the proximal centriole of sperm. So two things that enter inside the ovum is male pronuclei. Why you are calling it as pronuclei? Because they are just going to fuse. Pro means before nuclei, right? This is not the one that is going to be formed. Or we can say these are the nuclei that are separated, or these are the nuclei of male and female separately present or prior to the fertilization, okay? And this one is female pronuclei. Now the fusion or syngamy will take place, karyogamy will take place. There are two processes under fertilization: syngamy, fusion of plasma membrane, that has already occurred, and karyogamy, that is a fusion of nuclei. And now the last step of fertilization is karyogamy. Karyogamy means, karyo word is always used for nucleus. That's the fusion of pronuclei. Alright, for example, this is your ovum. It has its own nucleus. Now both the nucleus are going to form, this is N, and they will form a diploid zygote. They will form a diploid zygote, and this zygote will now undergo divisions, and which will be talking about that in next class. Okay, so this is how the entire fertilization, guys, takes place. Okay, easy peasy. Now one question that you must be thinking, or many of you must be thinking, that ma'am, a lot of sperms are entering, but why only one sperm always fertilize the oocyte? Or why not one or two sperms can fertilize the oocyte? What's the reason? Let's see. So the term that we use is polyspermy. Block to polyspermy. We don't want polyspermy to happen. What is polyspermy? When more than one sperm fertilize the single oocyte. For example, this is oocyte and number of sperms are fertilizing. If number of sperms are fertilizing, this will become 3n, and 3n zygote cannot survive. It cannot survive, right? So we want that this polyspermy should be prevented. So there are certain blocks, or we can say what are the mechanism that does not allow the polyspermy to occur. For example, I say in ejaculation 200 to 300 million sperms are present, and if that man is perfectly fine, healthy, 60% should be in proper shape, and of that 40% will have the vigorous motility, and that's huge. And only 100 to 200 reaches fallopian tube, but 100 to 200 is also so much, right? Then why only one is able to fertilize? So for that there are certain reactions that goes on. So first block to polyspermy is fast block. Fast block to polyspermy. So what happened in fast block? For example, this is the vitelline membrane. Okay, so as soon as the first sperm will touch the vitelline membrane or plasma membrane of the oocyte. This is the first sperm. Imagine this is the first sperm. Okay, so as soon as the first sperm will touch the plasma membrane or vitelline membrane of the oocyte, the entire membrane will undergo depolarization. You understand what's depolarization? Remember, we have done the depolarization and repolarization like this. Yes, the inside becomes positive, so that's depolarization. Earlier it was at rest. Earlier it was like this, inside was negative, outside was positive, right? When it was at rest, it was like that. So when the sperm will touch the membrane, when it is at resting position, what will happen after that? Due to touching a stimulus will arise to the membrane and it will undergo depolarization. Now what will happen if it undergo depolarization? Because it is occurring very fast, as soon as it will touch, it will undergo depolarization. So that's why it is fast block. But what will happen with depolarization? See, every sperm's membrane have certain proteins known as voltage sensitive protein, and these voltage sensitive protein can only sense this much of resting membrane potential, which is the resting membrane potential. Okay, it cannot judge depolarization, right? For example, the first sperm that enters, it has seen the resting membrane potential of around -70 millivolt. And this is appropriate for the proteins present on the surface of the sperm. And now the sperm has entered inside the cytoplasm. Now second sperm will come and touches this membrane, and this will get a shock and it will then move away from the surface. So basically, first sperm that enters, it will generate a current on the membrane, right? And that current shock will occur to the second sperm, and second sperm will not be able to enter inside. So in short, I will say the sperms have certain proteins on the membrane which can you can say, or these protein can only judge the resting membrane potential of this one, not the depolarization of +30 millivolt. If the sperm get this resting membrane potential, only then it can enter. If it will get this one potential, it will not be able to enter inside. So this is how the rest of the sperms will not be able to move inside. So let me write this for you in detail. The first sperm touches the plasma membrane of oocyte. Then what will happen? Depolarization. Right? So this is a membrane, guys. This is a membrane, plasma membrane. We always say that outside it is positive, inside negative when you are at rest, and after depolarization the reverse polarity occurs, right? So when the depolarization occurs, now what will happen? No other sperms can enter inside oocyte. Right? What do they have on their surface? They have voltage sensitive protein. They have voltage sensitive protein. They can judge the voltage. They can only be familiar with -70, +30 is like a shock for them. So these proteins will be destroyed. Fine. Now that's a fast block because it occurs fast, but it remains only for small duration or little duration. If it occurs only for little duration, maybe for seconds, it can block the 2 sperm that come simultaneously, or 3 sperm or 4 sperm that occur simultaneously. But what if the sperm is or what about the other sperms that are entering? For example, in single second number of sperms are entering. For example, first sperm is this and second one is just behind this one. The fast block can block those sperms, but after few seconds the fast block will be vanished. Then how will you prevent the entry of the other sperm? For that we have slow block. For that we have slow block, also known as cortical reaction. So what happens in slow block? See, this is a vitelline membrane. This is a vitelline membrane and this is your zona pellucida. This is the zona pellucida. Let me label this for you. This is the vitelline membrane. This is the zona pellucida. There is a space present between vitelline membrane and zona pellucida. We call this as perivitelline space. What do you call it as? Perivitelline space. Now we suppose that the sperm has already entered here. The sperm has already entered here and fast block has happened. The depolarization of the entire membrane has taken place. So here depolarization has taken place. By this depolarization, imagine depolarization has taken place, okay, of the entire membrane. Now, due to this depolarization wave, the calcium channels will open. What will open? Calcium channel, and calcium will move inside. So this calcium is moving inside the oocyte. Okay. Now, this calcium will fuse with cortical granules. What are cortical granules? These are very small grain-like structures, these very small granule-like structures present on the periphery of the oocyte, right? Not in the center, on the periphery. So these cortical granules, let me make it here. These cortical granules are present at the surface, and these granules, they will fuse with calcium ion. They will fuse with calcium ion. So there is fusion of calcium ion with cortical granules. So when these cortical granules, they will fuse with the calcium ion, what they will do? They will start moving inside the perivitelline space. So you can see here, they will start moving in the perivitelline space. After fusion, the cortical granules will move in the perivitelline space. Let me tell you one more time. After slow block, the depolarization wave that occurs, it will increase the concentration of calcium ion inside the cytoplasm of oocyte. This calcium ion, they will fuse with this cortical granule. These granules, when they will fuse with calcium ion, they will move in the perivitelline space. After they move in the perivitelline space, they starts releasing enzyme. So these cortical granules have a purpose. They release enzyme. They release enzyme. These enzyme, they will destroy the zona pellucida or they will alter the zona pellucida. Okay, earlier zona pellucida was smooth. Now zona pellucida's protein receptors have been destroyed. If their protein receptors are destroyed, the ZP receptors, the sperm will not be able to recognize the oocyte and sperm will not be able to enter. So the major purpose here of the slow block is to permanently change the ZP receptor. What they are doing? They permanently change the ZP receptors. If the ZP receptors are destroyed by the enzymes of cortical granules, then what will happen? The other sperms cannot enter because there will not be species recognition. So that's the permanent thing. So we have permanently destroyed the ZP receptors so that no other sperm can enter. So this is how only one sperm can easily fertilize one oocyte, right? So this is how the slow block by giving current, fast block by giving current prevent other sperms to enter. This is just like, for example, you enter into the room via door, and when you will shut the door will start having an electric current, and when the second person will touch the door, the person will get electric current and the person will fall off. Just like this. The first one that will come, the door will be normal, and when the second one will come, it will get an electric shock because due to entry of first person, the first person shut the door and it got the electric current. Okay, earlier the door was open and now you entered, you are the first person, now you have closed the door. Now door has got the electric current, and when the second will touch it, it will fall off and it will not, or that second person will not be able to enter inside the room. Okay. Now, what about slow block? Due to the wave of depolarization, cortical granule, they will fuse with calcium ion. Calcium ion has entered due to depolarization. Now, this cortical granule enter into perivitelline space, and then what it will do? It will release enzyme which will destroy the ZP receptors of zona pellucida. Hence, no other sperm can enter inside the oocyte. Fine. That's about the fertilization and prevention to polyspermy. Let's talk about sex of the baby. So now, as you all know, the ovum produced is always having total 23 chromosome, which is 22 plus X. X is the sex chromosome. Okay, and the sperm can be of two type. The sperm can be 22 plus X and it can be 22 plus Y, right? So there are number of sperms that are entering in the fallopian tube. Okay, some are of 22 plus X type, some are of 22 plus Y type, but female is producing only one egg and every time it will be containing X chromosome. Okay, now there is a probability that X chromosome or the sperm with X chromosome has fused with the ovum. Then what will be the baby? For example, that's the zygote formed by the fusion of these two gamete. Then it will be 44 plus XX, and XX tell us that this will be a girl child, right? Now if the fusion is in such a way that the same ovum is, or we are taking second probability, that what if the sperm which is going to fuse with the ovum is 22 plus Y. Now the zygote form will be of another type. It will be 44 plus XY. This will be a boy, right? Because boys have XY chromosome, girls have XX, right? So from this, what we got to know? We got to know that the female who is producing egg has no control on the sex of the baby. The control on the sex of the baby is in the male's hand, not in the female's hand. The female has no role in the sex of the baby. The only role is of the male, because male produces two types of gametes and female produces only one type of a gamete. Fine. So you know, even in India, in villages of India, there is a thing that still females are blamed to give them a girl child, right? Right now girls and boys both are equal. It's nothing like that. I'm a girl and I'm teaching you. Might be on somewhere else the male teacher is teaching the zoology. Then what's the difference? The only difference is I am a female, I have X chromosome, I have female reproductive parts, and male have the male reproductive part. That's the only difference. I am also teaching, the person is also teaching. I have my other qualities, he might be having some other qualities. I am also supporting my parents, my family. The male person is also supporting the parents and family, right? So female is nowhere different from the male nowadays, right? And it's a myth that females are inferior and something like that. It's not like that. It's just a matter of your vision, you know, how you see both the genders. That's it. It's about on your own thinking, right? So the female should never be blamed for the sex of the baby, or the male is responsible for the sex of the baby. In fact, he is also not responsible. He is only producing 2 types of gametes. Even he doesn't know which is going to hit the ovum, right? So it's a chance process. It's a probability. So that's about the sex of the baby. Let's read NCERT. During copulation, which is coitus or sexual intercourse, semen is released by the penis into vagina. You call it as insemination. The motile sperm swim rapidly through the cervix, enter into uterus and finally reach the ampullary region of fallopian tube. The ovum released by the ovaries is transported to the ampullary region where fertilization takes place. So it's clearly written where the fertilization takes place, ampulla. Fertilization can only occur if the ovum and sperm are transported simultaneously to the ampullary region. This is what I have told you. If both they meet at one particular point at same time, only then fertilization will occur. This is the reason why not all copulation lead to fertilization and pregnancy. So because it's a probability, so not all fertilization can lead to pregnancy, okay? And so this is always you know, in the movies it is always shown that there is one night stand or sexual intercourse and the female got pregnant. It's not in all the cases. This can also be true. This is, I'm not denying. This can also be true that in just one sexual intercourse a female gets pregnant, but it's not in the majority of cases. In majority of cases, not all the copulation leads to fertilization. The process of fusion of a sperm with the ovum is called fertilization. During fertilization, a sperm comes in contact with zona pellucida layer of ovum and induces change in the membrane that block the entry of additional sperm. So NCERT has not mentioned these blocks in detail, but yes, they have mentioned when it comes in contact with, the sperm get contact with zona pellucida and induces changes in the membrane that block the entry of additional sperm. They are talking about slow block. Thus, it ensures that one sperm can fertilize an ovum. The secretion of the acrosome help in the sperm enter into the cytoplasm of the ovum through the zona pellucida and plasma membrane. We have done acrosomal reaction, how various enzymes are helping in penetration of sperm. This induces the completion of meiotic division of secondary oocyte. The second meiotic division is also unequal and results in the formation of second polar body and haploid ovum. This we have already discussed in oogenesis. In female, the divisions are always unequal. That's why there is very small polar body and a large ovum. Now, ovum egg ooted. Sorry. We have done in the previous class. They are all same. They are just another names. Now, soon the haploid nucleus of the sperm and that of a ovum fuse together to form a diploid zygote. How many chromosomes will be there in the zygote? It's 2n. So 44 plus XY or 44 plus XS. Total 46 or 23 pairs. One has to remember that the sex of the baby has been decided at the stage itself. So during fertilization only you get to know what is going to be formed, the male or female, because which sperm will enter, that will decide, okay. As you know, the chromosome pattern in the human female is XX and that in the male is XY. Therefore, all the haploid gametes formed by female have the sex chromosome X, whereas the male gametes, in the male gamete, the sex chromosome can either be X or Y, right. So I have told you the male can produce this and that, female will always produce X. So, probability of getting X chromosome in female is 100%, but in male is 50-50%. So, this is going to be your next line. Hence, 50% of sperm carry the X chromosome while the other 50% carry Y. After fusion of male and female, the zygote would carry either XX or XY. So, any one of them will be in the zygote, depending upon whether the sperm carrying X or Y fertilize the ovum. The zygote carrying XX would develop into female and XY would develop into male. That is why scientifically it is correct to say that sex of the baby is determined by the father, not by the mother. Alright, okay. So, let us solve the question. Which of the following enzyme helps sperm to penetrate zona pellucida? Hyaluronidase, neuraminidase, acrosin, corona penetrating enzyme. So, first is hyaluronidase. This is the enzyme that digests hyaluronic acid. So, that is not the answer. We want the one that digests zona pellucida. This is the one we have not discussed even yet. This is out of your even the syllabus. This is an enzyme that digests a carbohydrate in your plasma membrane, right? So, this is not the answer. Acrosin or zona lysin is the one that digests zona pellucida. And corona penetrating enzyme, it helps to penetrate through the cells of corona radiata. So, answer is 3. Next, which centriole of spermatozoa is required for first cleavage? Proximal centriole, distal, ring centriole, posterior centriole. This is very straightforward question. There is only one centriole that enters inside the ovum, the only one centriole of the sperm, and that is your proximal centriole. So, answer is 1. And as I have told you, it helps to make the spindles. So, that will complete the first division, okay, and that will also be discussing how it happens in the next class. Next, which of the following option is correct for the label part A, B, C? So, this is the diagram given in NCERT also. I have this diagram, right, that is the same diagram. So, as I always say, always pay attention to diagrams, okay. So, here you can see these are the cells of corona radiata, zona pellucida, and that is your vitelline membrane, and this space is perivitelline space, okay. So, let us see what it is saying. So, this is cells of corona radiata. This is perivitelline space, this is zona pellucida. So, A is not follicular cell, A is zona pellucida, B is cells of corona radiata, and this is perivitelline space. So, answer is 3. Next, why do all copulation not lead to fertilization in pregnancy? The root causes. There is only one reason why all pregnancy do not lead to the or why not all copulation lead to the pregnancy, because both the gametes have to reach on the right time at right place. So, what will be the answer? Read the options. Due to numerous sperm in one ovum? No, that increases probability. The increase in if one gamete is even increased in number, that will increase the probability. So, that is wrong. Not due to hormones, hormones have no role here. Ovum and sperms are not transported simultaneously to the ampulla, very true. Due to non-formation of corpus luteum, as I have told you, no role of hormones, no role of corpus luteum. So, this is true because both have to reach at ampulla simultaneously. Next, what induces the completion of meiotic division of secondary oocyte? Okay, what is that force that leads to the completion of meiosis II? So, meiosis II only gets completed when sperm enters the cytoplasm of ovum. Contact of the sperm with zona pellucida? No. Entry of sperm into the cytoplasm of ovum through the zona pellucida and plasma membrane. Entry of sperm in the ampullary isthmic junction, that is incorrect. Copulation? No. So, this is the answer. It should enter into the cytoplasm, because when it enters the cytoplasm, the metabolism of the cytoplasm of ovum get boosted up and completing the meiosis II. Next, why it is scientifically correct to say that sex of the baby is determined by father, not by mother? Human female produces 2 types of gamete. Human female produces 1 type of gamete. Human female is XX, male is XY, 50% of sperm carrying X chromosome, while 50% carries Y, all of these. So, female produces only 1 type of gamete, male produces 2 types. So, these are wrong. So all of these will not be there, and this is true. Female produce only XS, male produces both X and Y chromosome, which can or which are responsible for either boy or a girl. Next, fertilization in human occurs at, very simple, ampulla. Answer 2. So that's it about fertilization. In the next class, we'll be discussing about cleavage, implantation and other topics. So stay tuned, keep on working hard, complete this one before the next class, and I'll meet you in the next class. Bye-bye.